You can convert atmospheric nitrogen and nitrate are often studied in what kind of microbiology?
Environment.
Yeah, environmental or soil microbiology. I think that's probably right. Yes, good job in the front. Okay. The blank of an organism is all the chemical reactions take place.
Metabolism.
Metabolism, good. Okay. Inserting a gene from the hepatitis B virus into yeast so the yeast produces a viral protein is an example of what? Good job, that's genetic engineering, putting DNA from one organism to another and getting a protein. Excellent. See, this is the perfect level for you guys, right? Who discovered penicillin? Anyone know?
Fleming.
Fleming. Alexander Fleming. Good. We'll get to all those other guys over the class, though. Where am I at? Okay, we're more than halfway now. The term for the use of microbes to restore damaged environments is? Bioremediation. Good. We studied it the first day. Production of human blood clotting factor by E. coli is an example of bioremediation. Now that would be genetic engineering, right? Bioremediation is when you clean up something. Okay, after using an inoculating loop or needle to transfer a culture from one medium to the next, what steps should next be taken to avoid contamination? Yeah, red hot, right? You don't just wipe it with a paper towel, clean it with soap and alcohol. Duh. Okay, but I'm going to have laboratory safety on the test too, so that's good. What's incorrect? Eating and drinking is allowed as long as open culture. Ooh. See, technically we should have no eating and drinking, right? Yeah.
The question is false.
Which one's false? So we should not allow eating and drinking. Well, we want a false question, incorrect. You don't want to put cosmetics in the lab because you're touching stuff. Get an eye infection. I have to think eating and drinking is not allowed, technically, right? So that's what they want. I let you guys eat and drink because I'm soft on you. Okay. What's an example of proper laboratory technique? Lick labels before adhering to culture tubes. Place cultures into the trash can. Are we going straight trash? Pipette by mouth only.
That's the last one.
Duh, my God. Do we do mouth pipetting here? Microbes? That's funny stuff.
Did you make these questions up?
No, they're from a test bank. I just picked them. Okay. But I thought we were all going to be able to do them, submit them. It was going to be much better. Just didn't work out. What anchors the bacterial flagellum in the cell wall? It's not the pili. No. There's a word. It's two words actually. B and a B word. And the second word is body.
Basal body.
There we go. I sprang a brain cell. Okay. It's part of the flagella that holds it into the wall. Basal body. Bacterial pili can be described as what? Ooh, that's hard. Yeah, I think there's special fimbriae. They're not peptidoglycan, that's the cell wall. They're not really locomotion, they don't use that. So we can knock out those two. Are they rigid protein structures? Well,
No.
It's holding on to the thing.
so you have three answers there that are not so bad. I think we're going to submit the one I got. I knocked out 2 out of the 5. And we got it right. Okay. Tubulin is part of flagella, and I guess they're not solid and rigid. They're softer, stickier. Okay. Which of the following bacterial cell structures plays an important role in the creation of biofilms?
Oh no, glycocalycis.
Right, biofilms are the sticky secreted material. So I know glycocalyx is part of it, but is fimbriate part of it? I don't think so, but maybe they have a different idea about that. Well, should we try that one?
Yeah.
We always have more attempts left. Okay, remember glycocalyx is a sticky layer. Biofilm is a bunch of bacteria in a community with a sugar layer around them. Now, do the fimbriae play a role? I'm not sure. Nope, but they might. Let's try. Yeah, it's both.
Can you go to the
Okay.
root part?
And the root part's still blue.
Oh.
Yeah, there's not much view material here. Biofilm and fimbriae. Yeah, fimbriae enhance the biofilms. Cool. And allows them to stick together better. Cool. So they are part of this. Cool, I should have done that. Bad teacher. Which of the following cell walls contain teichoic acid?
Gram-positive.
Yeah, it's the gram-positives, okay? So, gram-negative don't have that. Good. All right, how far are we? 34? We got 16 left, 15 left. Are you with me, people?
Yes.
Okay. After this, we're gonna have a break, and then we'll have individual time after that for tutoring. Okay, does that work for you?
Yes.
15 more questions. We'll do a tutoring between 4:30 and 5:30, if you need it. Okay, starting to cough.
Gram
What contains an outer membrane?
Gram negative.
Gram-negs. Okay, do the archaea have an outer membrane? I don't think so. Okay, we're good. Correct. Okay, 14 left. What's not a component of cell wall? Yeah, tubulin, that's part of flagella. Okay. The rest are part of the cell walls. Okay. Lipid blank is part of the gram-negative cell wall, can produce fever, inflammation, and shock when it's released into the bloodstream. What's it called? Lipid A. Yeah. Lipid A is part of the LPS layer. It's the toxic part. Okay. Which of the following chemical substance contributes to the unique changes of acid-fast bacteria? The ones that contain the first stain you give them in the acid-fast stain. What kind of bacteria are acid-fast? Mycobacteria. Mycobacteria. Okay, so mycolic acid. That's why they call it mycobacteria. Look back in your notes, it's there from the lab. Okay? What mycobacteria are. In a blank solution, animal cell can gain so much water that it may burst.
Hypotonic.
Yeah.
Or high.
No, in a hypertonic environment, they'll shrink. Hypotonic, they're gonna fill with water and burst. ATP is expended in which of the following processes? Ooh, good one.
Active transport.
Let's find out what group translocation means. Where'd it go? Group translocation. Is there going to be an animation here? Cool.
Group translocation is a process in which an organic molecule, such as glucose, is transported into the cell while being chemically modified. The PEP phosphotransferase system is an example of group translocation.
PEP.
In this process, the phosphate from the high-energy compound phosphate